Today I'm going to present an ongoing line of research that I studied several years ago, started several years ago, that started from a naive observation looking at RNA sequencing data, which develops to a huge snowball, which challenges some of the most fundamental concepts in transcriptome and proteome studies. So in 2003, when the Human Genome Project has finalized, one of the main thing that researchers were surprised is the low number of genes in the human genome. While we were expecting to have around 100,000 genes in the human genome, they saw only about 20,000 coding genes. And later on, when they start to map the genome of even lower organisms, that question became even more strange, because, for example, for C. elegans, which is only like 1,000 somatic cells, has more or less the same number of genes as the Homo sapiens. So if we go to the central dogma of how a gene in the eukaryotic system looks like, the gene structure is basically built from several elements: the coding sequence, the 3' UTR, 5' UTR, and poly A and cap. While the coding sequence and 3' UTR can be remodeled or changed by alternative polyadenylation and alternative splicing, the cap considered to be as an anchor for the messenger RNA molecule. So several years ago, I started to notice abnormalities in RNA sequencing that could not be explained by the central dogma. To make a long story short, what we found was that when alternative polyadenylation happens in the 3' UTR, it's not only terminating the upstream RNA, messenger RNA fragment, it's also generating an uncapped, stable 3' end of RNA. And these fragments that mistakenly considered to be a part of the 3' UTR are in fact an autonomous RNA fragments. And from this point further, I will call this fragment as a 5' uncapped polyadenylated transcript, or in short, 5' UPTs. So on top of all what we did in this research, one of the insights that I had following that study was that the metazone transcriptome is much more complicated than has been thought. And when I started my postdoc at the Netherlands Cancer Institute, I remember sitting one day and looking at just the genome part of some random genes, and I suddenly realized that I'm actually looking at something that cannot exist in the biology world that we actually know. And I will explain. If you see here for these two random genes, there are multiple variants that cannot be explained by either alternative splicing, since the first cap exon must be included during any combination of splicing, and it can also cannot be explained by alternative promoters, since there is only single promoter at the 5' end. So what I was thinking, that what I discovered before in the 3' UTR, that the 5' UPT can be cleaved to generate an autonomous uncapped RNA, also happens in the coding sequence region. So we decided to take this observation several steps forward and doing on a transcriptome level. So based on the distinctive molecule characterization of the uncapped and the capped RNA fragments, we've been able to isolate the cap RNA by cap IP or by exonuclease treatment called TEX to degrade the uncapped RNA, and by doing that, enriching the cap RNA, or by 3' pull down of the uncapped RNA using in vitro capping system with bioattenuated GTP. And this is just a random gene to show you how the data looks like, and this is the total RNA of the control. The cap IP enriches the 5' end and also the TEX, while the 3' pull down enriches the downstream uncapped RNA. To analyze the data, we use the hidden Markov model to identify the most probable cleavage point, and in addition to that, to quantify the amount of uncapped RNA for every gene. And the result was kind of ground shaking. What we found was that in the transcriptome, the human transcriptome, around 7,000 genes has about 30 to 50% expression of 5' UPTs, which means that beneath the layer of what we know as the capped transcriptome, there is a highly enriched uncapped transcriptome that we simply don't know and we're not starting to uncover. So just a quick characterization, molecular characterization of 5' UPTs. So we found that in line with the previous study, that the endonuclease cleavage and polyadenylation plays a critical role in generating the 5' UPTs. We found that 5' UPTs exhibit highly structured RNA at the 5' end, which protects them from exonuclease like XRN1 or XRN2. And this is also kind of shattering 50 years of dogma that uncapped RNA are highly unstable. And the last thing that we saw was the 5' UPTs are marked with 5' and M6A modification. So in recent years, there are hundreds of publications that show the involvement of M6A metabolism in RNA structure, stability, splicing, alternative polyadenylation. But I want to refer something that we found might be very relevant to our study, which is a study that has been published in 2015, where they show that when M6A is located on the uncapped RNA at the 5' end, it can promote cap-independent translation. So we had the thought that the M6A plus the uncapped RNA that we're seeing may generate translation in the cap-independent manner. So to address this question, we had to think outside the box how we can show it proteomically, since regular mass spectrometry will simply lose all the critical data, because when we give a sample to mass spectrometry, one of the first steps that you do is to a proteolytic process that degrades all the proteins into peptides. And by doing that, we're basically losing all the critical information or the hidden information that we're looking for. And with a collaboration with the Lee Lab from McCune University in Seoul that developed an N-terminal mass spec, we've been able to mass spec only the N-terminal of the entire proteomics in mass spec. And by doing that, we can identify if the peptides indeed derives from a canonical protein or from non-canonical one. And we joined this data to 5' uncapped RNA sequencing data that uniquely sequenced the 5' end of uncapped RNA and an M6A data. And as you can see here, what we saw is a very strong overlap between the three different methodology, which implicates that indeed the 5' UPT is extremely enriched transcriptome, let's say, layer that contribute to the proteome in a cap-independent manner. So if I had to summarize the model till now, what we found basically is that for thousands of genes, when endonuclease cleavage based on alternative polyadenylation cleave the RNA, it can generate a new transcript with structural RNA that protects it from exonuclease and promote cap-independent translation to generate entirely new protein. And if I had to summarize it, the mammalian genes that are, seems to be highly enriched microenvironment that can produce multiple mRNA protein variants in a polycistronic way, very similarly to the bacterial system. So if up until now I was focusing on the what and the how, by the way, in the manuscript, upcoming manuscript, welcome you to read, we also show some functional with the 5' UPT and the immunopeptidomics and the regulation of BCL2. But I would like to focus my last two slides in the general overview of the why, or the project that I'm working now to follow up what I did till now. What is the biological function? So in recent years, there are hundreds of publications that's showing highly dynamic alternative polyadenylation, M6A methylation, cap-independent translation. And even though they were working independently, like independent field, there is some correlation that has been shown between these different fields, especially they're going up upon stress, mitosis, differentiation, T-cell activation, multiple types of cancer, and more. And my hypothesis was that what we discover now as a new class of RNA as a 5' UPT biology may be a critical piece of the puzzle that can complete an exciting new biology that was hidden within the CAP transcript. So to make a very, very long story short, what we start to unravel now when the dust starts to fall down is that if alternative splicing is the major dogma in the field that can generate the diversity, can increase the diversity between genes, which is like the fine tuning of many genes, we found that the 5' UPT is actually a way to sub-regulate a special domain or specific domain in a protein, which means that the genes are actually a modular system. They're not working as entire sets of domains that can actually work independently as independent domain, which can affect the function, localization, interaction protein complexes, et cetera, et cetera. And what is even more striking is that in a certain condition, this happens in thousands of genes in parallel. One of the things that I'm currently working on is the T-cell activation, where we see a huge difference in thousands of genes. And I just want to give you one small example of JAK2, which is a major kinase in T-cell activation that responsible for the JAK-STAT pathway. And as you can see here, the JAK is basically assembled for several domains. This is the kinase domain in the C-terminal, and upstream to it is the JH2, which is a pseudokinase that inhibits the kinase domain and its activity. And what we saw is that upon activation, the RNA is being cleaved just between the JH2 and JH1 domain. And what happens, as you can see here in the T-cell activation, this is the total RNA and polysome fraction. And this is the Western blot, it's in the macrophage activation. It can releases the kinase domain to now act freely without its internal inhibitory domain. And I just want to thank Reuven Agami, my PI, for supporting me and letting me do what I do, and all the collaborators and our facilities, obviously our sponsor,